Hello everyone, welcome back for another session of regeneration biology. So in the last class, we did not complete the mechanism. So hence I have given two titles. One is the mechanism of regeneration in Hydra, which is continuing. And then comes the asexual reproduction, where we discuss the fragmentation, morphallaxis, epimorphosis, and also a few lessons that you can learn from Hydra. So yesterday, in the previous class, what we have been understanding or trying to learn was that how Hydra make use of the various molecules for their regenerative to benefit their regeneration. So we will continue with the same section. So one gene that is called pedibin, which is needed for specifying the food, as you can see here, and it is expressed both in the food of Hydra and also in the base of the tentacles. That means it is there near the head region and also in the base. And this, as you can see here, is in the B panel is the sense control RNA in situ hybridization. This is a normal antisense pedibin signal. You can see this is a higher magnification, and this is the base of the food. And you can see both in cross section or in whole mount in situ, you can see the pedibin is highly expressed in the foot and the tentacle bases of Hydra, and its expression of pedibin transcripts as detected using the whole mount preparations on cryosections and also using the digoxygenin labeled riboprobes. Riboprobes means RNA probes. They can go and bind because riboprobes will be the antisense probe. It will go and bind to the sense RNA probes, and later you can detect this digoxygenin using the antibody, and antibodies will have alkaline phosphatase enzyme, and once you put the substrate for this alkaline phosphatase, you will end up getting a proper signal that will appear. So the food, as you can see here, it has got a heavy signal that is expressed, and also the base of the tentacles, you can see the signal, and the rest of the bottom panel, it is the on cryosections. Those who want to know more detail, you can see this DOI URL given. It is from a published paper. So another peptide, apart from this, there is another peptide that is known as HYME323, and which is a 16 amino acid long peptide that shares no structural similarity to that of HYME346, which we discussed in the previous class. Is expressed both in the ectodermal and endodermal epithelial cells throughout the body, except for the basal disc and the head region. So this HYME323 is capable of providing positional identity. It is expressed both in the ectodermal and endodermal epithelial cells, and it is contributing for the positional identity. And when you do the transplantation and regeneration experiments, it indicates that upon initiation of food formation, the stored HYME323, that means it's a peptide, the 16 amino acid peptide, the stored HYME323, it will be released from the epithelial cells and induces differentiation of the basal disc cells of the food. So just like if you have got a wound in your hand, if it is exposed to the air, it will clot. But before that, the platelets explode by contact with the air, and it will release a factor called PDGF, platelet derived growth factor, which go and bind onto the receptors of PDGFR, which is present in every cell, and that will stimulate the cell proliferation. So same way, HYME323 also stored in the cells, and whenever there is a insult that occurs to the cell, it will be released and it can kickstart the feed formation, not just the cell proliferation. Same way, another protein called anklet, which is a protein. Anklet means, I guess all of you know, where ladies will wear as an ornament in their feet. It's easy to remember. Anklet is a protein with a perforin and EGF domain. EGF is epidermal growth factor domain, which mediates food specific differentiation. Now, the previous, whatever this factor, HYME323, etc., can initiate the food formation, but they cannot make the food. So it's just like, you purchased a land and to make a house, but land is not house. Land is a place where you can build a house. So someone has to build a house in order to start staying. So this anklet makes sure that this destined area now will give rise to differentiation of these cells into these proliferating cells into food specific morphology. So anklet protein is functionally involved in the transition of the ectodermal epithelial stem cells into food specific basal disc epithelial cells. Anklet, which has a signal sequence at its end terminus, and it has got two domains that are called MAC PF domain. MAC stands for membrane attack complex and PF stands for perforin domain, and it also contains EGF domain. What does signify these domains? They can interact with other proteins and attract other proteins which will interact with these domains and can drive the gene expression events in a specific manner. So in a food regenerating hydra, anklet is first expressed in the newly differentiated basal disc. Basal disc initiation happened because of the peptides what we discussed earlier, and the basal disc cells now start expressing the anklet cells at the regenerating basal end, and then the expression becomes restricted in the lowest region of the peduncle. You don't want other parts of food to form from in the body, belly region or head region. You don't want. You want food should be in the food. You don't want any other place. So that's why the anklet expression is very much restricted to the extreme end of the peduncle, where the animal's extreme end exists. So the suppression of anklet gene. What if you get rid of anklet gene, knock it down? What will happen? Led to a smaller foot. Foot doesn't look like a foot, and significant decrease in the basal disc size. So this would be, say for example, your foot may be say 30 centimeter or 20 centimeter long from tip of your toe till your back of your feet. What if it is made into like that of a small baby? You will struggle to balance, right? So food has to have a specific size if it has to anchor. So decrease in the basal disc size, and during the foot regeneration, there is a delay in the basal disc regeneration. So if you knock down or suppress the anklet function, you will end up getting a deformed or malformed foot region, either during regeneration or the structure that is being formed from a newly formed hydra. So regeneration is via communication. That is, signal transduction pathways in hydra regeneration are very much important to discuss. Without cell-cell communication, regeneration would not happen. Cell-cell communication basically means if one cell is not able to communicate to the other cell, like you have seen similar mechanism in the liver regeneration. If one cell is, one hepatocyte is priming the other hepatocyte. If that doesn't happen, the regeneration also cannot happen. So hydra regeneration communicate exogenous signals to the transcriptional machinery. Exogenous means something released from a cell acted on another cell's receptor. Say, classic example is Wnt beta-catenin signaling. Wnt bind onto the frizzled, and the beta-catenin gets stabilized inside the cell and it goes to the nucleus and start its action. So these signals must get translated into a recipient cell that will influence the transcriptional machinery. Hydra's regeneration tissue shows remarkable conservation to the signaling pathways used by the developing vertebrate embryo. Vertebrate, which is highly advanced organism. Hydra, in comparison to a vertebrate, is very primitive in terms of organismal complexity. But the signaling mechanisms are very much similar to that of a developing vertebrate embryo. One example is BMP 5 to 8. BMP stands for bone morphogenetic protein, which is a very predominant signaling event that occurs in every organism's development. And BMP5, BMP6, BMP7, BMP8, these can be activated, the respective receptor target. BMPs and TGF-beta are somewhat like that of cousins, or I won't say sibling, but you can compare them to that of cousins. Cousins are genetically related, but they are not as related as that of siblings. Okay, so these orthologs of BMP 5 to 8 and the member of one of the most complex group of cytokine superfamilies that consists of the TGF-beta isoforms and other family members such as activin A. So activin, nodin, BMP, these are all required for the activation of various regenerative as well as developmental pathway. And we have orthologs. Ortholog means you are having a gene identified from an organism. You are trying to identify a similar, functionally similar gene in another organism. Not structurally similar. Functionally similar, that we often call it as ortholog, but they can have structural similarity also. So there are BMP 5 to 8 orthologs present in the Hydra. So regeneration via communication, when it happens, this Hydra BMP 5 to 8, that is what is high BMP 5 and 8, is activated, and that is essential for the tentacle formation. Tentacle, if a new head is formed in a decapitated Hydra, new head is formed, it should have the tentacles also. And tentacle formation is dependent a lot on this Hydra's specific BMP 5 to 8. And in patterning, the lower end of the body axis. In the body axis, when it has to form, the lower end specification also is contributed by the BMPs, Hydra BMPs. BMP antagonist. Antagonist and agonist are common terms used in every cell signaling event. So antagonist is something which is opposing it. Say you want to speak and someone encouraging you to speak is a agonist. Someone who is telling not to speak, or someone put a cotton in your mouth, and they are antagonist. So this is the signaling pathway. So BMP has an antagonist called gremlin, which is a cysteine knot protein that is present abundantly in vertebrates that antagonizes preferentially BMP2 and BMP4 on an average. Is one of the factors involved in head regeneration. In Hydra, for head to regenerate, you need to have the BMP antagonist also. Like I already told you, if you want to drive a car, you need to have not just accelerator. As important as accelerator is the brake, because driving is not a one-sided affair. You want to stop driving also. If you cannot, then you will end up getting accident. Same way, this antagonists do as important job as an agonist or the activator itself. Another putative cytokine in Hydra is genes encoding insulin-related protein. Remember, Hydra also have got protein similar to that of vertebrate insulin, which led to the identification of 3 candidate genes for insulin from Hydra, which we know from the insulin-related. Insulin-related gene basically means it is not exact insulin, but it is mimicking the function and it's mimicking the role of insulin. Even we have insulin-like growth factor, we call it as IGF. So the identification and characterization of the Hydra insulin receptor, which is called HTK7, it supports this view that the Hydra use insulin signaling. Normally, insulin signal is not just for absorption of the glucose into the cell, but it plays a major role in the cell proliferation, also in various reprogramming stages. Insulin is a multifaceted signaling molecule. So that also contributes in the regenerative events of the Hydra. Now let us see what is morphallaxis and epimorphosis, which is extensively discussed in various stages, but we will revisit them once again. Any isolated fragment of the Hydra body which is larger than a few hundred epithelial cells, any part of the body you take and you just smash it. Smash means not break the cell, just decimate them into a few hundred epithelial cells, can regenerate into miniature version of the new animal. So we can call this as a asexual reproduction. Asexual reproduction means there is no involvement of gametes. Although Hydra is capable of sexual reproduction, it can make a new Hydra from a small tissue or a few hundred cells from its body. And this process is called morphallaxis. Morphallaxis refers to the type of regeneration that occurs in the absence of cellular proliferation and involves the transformation of the existing body parts or tissues into newly organized structures. Say you took a small portion of the Hydra from its belly region. That means belly region doesn't have head, doesn't have foot, doesn't have tentacle, or doesn't have many other organs. But these 100 cells, it will be arranged in such a way that it will form a miniature Hydra, very small Hydra. So that means the belly cell will be now distributed as a head, as a tentacle, as a foot, etc. It is repurposing, although it is not formed by the active proliferation. 100 cell remain as 100 cell, but you arrange them in such a way that it forms a miniature organism. Now it will start proliferation. So this arrangement of the cells into a miniature organism without involving proliferation, this is very important, without involving cellular proliferation, that is called morphallaxis. But that miniature is useless. You can't do anything with this miniature organism. It's basically a model. It's like if you have a model of a Mercedes-Benz car, you can't put petrol and start riding. All you know is that it is a model. It is exact copy, but it's a miniature model. Many people will have miniature model of Taj Mahal, miniature model of Eiffel Tower, etc. They are not, but they mimic. You can put in your showcase. This animal, although it is live, it cannot perform the duties or job what a Hydra is supposed to do. In that situation, this has to now increase the size to that of a typical Hydra. How it is possible? By cellular proliferation. So first arrangement by morphallaxis, then you undergo epimorphosis. Epimorphosis refers to regeneration that requires active cell proliferation. So now you think about, you have got a tiny Mercedes-Benz car with you, very small one. Now it is suddenly becoming big, a big car size. Now you can put petrol, if engine and other things are there, you put petrol and you can ride on it. Something like that. That is what happens in Hydra's regeneration. Early regenerative processes in Hydra, however, always occur in the absence of DNA synthesis as the morpholactic process is powerful enough, in which the cells from the gastric region differentiate into head and foot specific cell. Just now I explained. Gastric region, say belly region, you take it, the same region cells, now it can give rise to head, food, whatever you want, it can give rise to. But it's not actually not that cell. It is basically mimicking. It is basically fooling around. However, in subsequent proliferation, the induction events are powerful enough that the foot will become actual foot, the head will become actual head, tentacle will become actual tentacle. It is not a knockoff copy or a non-functional, it's not a dummy or something. Now, morphallaxis and epimorphosis, if you see in the correct term, what we should know, when a Hydra is cut, the Wnt pathway is activated in the lower portion. Normally, the Wnt signaling gets usually activated in the portion that is in the bottommost portion of the body until it is capable of. So as soon as the head is cut, head is removed. So you have the body part, and there the tip of the body of the existing amputated body. So remember, Hydra is there, its head is cut. So now head is gone, that is not attached. The bottom, just below that head now, that is the topmost portion of the existing body. Hydra is cut, the Wnt pathway is activated. That means Wnt is somehow contributing. Lower portion, that is the portion that will eventually form the new head. So in other words, Wnt signaling is marking the place where the new head has to form. Without that, you cannot get a head. If the cut is made close to the head, that is just below the hypostome, Wnt3 in the epithelial cells will cause the remodeling of the existing cells to form a new head. That is, hence it is called a morphallactic regeneration. That means if the cut is made very close to the head, just below the hypostome, the Wnt can influence the epithelial cells and it will cause the remodeling of the tissue, existing tissue, and it will start forming a new head. That is morphallaxis to start with. However, if the Hydra is cut in the midsection, lower half, little bit lower to the head, the cells derived from the interstitial cells, that is where the neurons, nematocytes, gonad cells, etc. are there, they will undergo apoptosis immediately below that cut site. You made a cut, just below that they will undergo apoptosis. Why? Because you don't want those cells. You don't want neurons or nematocytes there. You want to make either head or foot in that region because it's an extreme now. Before dying, these cells produce a burst of Wnt3. Wnt is stored in the cells. In these cells, they are not released because of your mechanical damage. They rupture. Just like the release of the PDGF happened from the platelets of your blood. They release, and every cell that has got a frizzled receptor will bind with this Wnt3. So Wnt3 activates the beta-catenin in the interstitial cells. Actually, there is no plan of formation of head. Why now the plan started? Because of the wind. Who released the wind? The cells ruptured and they released the wind. Now they will start to make a head, causing a wave of cell proliferation. The interstitial cell that has far away from the head specific, they released. Now the neighboring cells are losing their identity and they start proliferating in these cells, as well as the remodeling of the epithelial cells. So it's now whichever cell has got a frizzled receptor, now it will undergo the beta-catenin stabilization and it will go to the nucleus and it can have the regeneration, and we now call it as epimorphic. So understand, in the first situation where the head is cut, the newly formed region, that is the rearrangement of cells, that is morphallactic. But whereas there is an active proliferation involved when you make the same cut towards the bottom region, not near the head but the bottom region, now these cells have to rearrange. Same wind signaling. Wind signaling is contributing in both the scenario, but in one scenario it is morphallactic, whereas little bit lower where you don't have any head specific signals that is existing, it will undergo epimorphic. It has to make lots of new cells. There is no head activator, there is no head inhibitor, it is far away from all this zone. And this property it makes use in its asexual reproduction, like such as fragmentation, which we will see in a short while. So Hydra is a small freshwater hydrozoan, which we have already seen, that are native to tropical and temperate regions. It is normally occurring in freshwaters and they don't seem to age or die of old age. Quite understandable, because their stem cells divide infinitesimally and they continue to divide, and because of this they have the ability to constantly replace the lost cell. If you constantly replace the lost cell, we can say that animal is immortal. Like one of the earlier classes we said, if an organism have extreme regenerative potential, we can call that organism as immortal because it is not dying. Like I told you, your skeleton's lifespan is 7 years. If every 7 years you replenish it, say you have no plan of replenishing, you decided not to replenish your skeleton, that means your lifespan is 7 years. Your skeleton disappeared means you are not there. Before that itself you will die. Maybe by 4 or 5 years you will die. You are not dying because your skeleton is being replenished. That means your body also have the ability to replenish. Like your skin gets replenished. In old age, people who don't have adequate skin stem cell, they start peeling off their skin. It will be almost like potato skin. You may have seen some newly potato coming in the market, you see its skin is peeling automatically, right? Like that, adult person, some people, not everyone, don't think everyone who is in 90s their skin will start peeling. Some people who don't have adequate stem cell, for some reason their stem cells are overworked. If you are working in sunshine, etc., you will have a high turnover, so your skin cells will be peeled off because you don't have enough stem cells to support. But if you have adequate stem cell, like in the case of Hydra, we can call Hydra do not die. Hydra reproduces asexually through budding and fragmentation. Two methods it uses. One is budding, which you can see in this picture. This is a full Hydra and you can see a small miniature Hydra that is being formed. And this also we have seen, this is the place where the head gradient and the foot gradient kind of nullifies. That is the place where it actually give rise to a new place. That means this region is neither favoring head nor favoring tail. So that area is a no zone area or doesn't belong to neither head nor foot. There the budding can happen. This we have seen in the earlier classes also, this picture. So here the budding occurs, and this is one of the most prominent asexually reproducing mechanism of Hydra. In both the processes, the offsprings are genetically identical to the parent. So any organism that is asexually reproducing, say a plant, you're taking a stick of a plant and you're planting. So basically you are making the clone of that animal. Clone in the sense genetical clone. Same way, if you are doing asexual reproduction, there is no way you will have any genetic variation, which is not very good for the animal. That is why Hydra has the ability to reproduce sexually also. So the budding normally happens, a bud forms on the body wall at, there is a spelling mistake, it is written and, it is at, should be read as at, at lower 2/3 region, and the bud grows into a miniature adult. As you can see, there is a miniature adult. The bud separates the parent body when it is mature. It will fall off and it will free float and it will go and settle down to one place. The new organism is a clone of the parent. Clone means morphologically it is same, but genetically it is identical. This is what we refer by clone. Now, Hydra is capable of doing another way of reproduction that we call it as fragmentation. Fragmentation means the Hydra can undergo damage due to various reasons. Like its body can get cut, maybe because of some sudden water movement, or maybe some predator. Predator means not an hunter or something. Some animal, smaller animal came and hit on it, etc. So it can break into pieces. So understand, the fragmentation means not that the Hydra get into fixed number of pieces. It can get into 2 piece, 3 piece, 4 piece, like that, random shape, randomly. The body of the Hydra breaks into 2 or more parts. Each part grows into a new individual. This is simple form, as you can see in this picture. There is a Hydra, and the Hydra, it cut into 2, forming fragments by loss of body parts. So the upper part lost its foot, the lower part lost its head. And now it arranged itself into a miniature Hydra, growing and regenerating fragments. So basically fragmentation is nothing but regeneration only, but since it's an asexual reproduction, it is often referred to as fragmentation. Now this Hydra grows slowly by cellular proliferation, and that will become a new Hydra, which is fully identical to that of the parent Hydra, genetically identical, and it can reproduce by budding, etc. It can also do sexual reproduction also by using the gametes. And this mechanism, asexual reproduction, is a process where an organism produces offspring without the need for fertilization. That means there is no egg or there is no sperm. But it is beneficial to sexually reproduce because it can bring in genetic variability. So that is why in a given environment, say one particular genotype of the Hydra is unfit, then any adversary, all genotype bearing Hydra can get wiped out. You would have seen during COVID also, not everyone had the same kind of mortality rate. Some people, not just with old or young, some people were more vulnerable. Even in some countries, people were more vulnerable because credit goes to their genotype. People who have got some specific genotype, which we cannot predict that you are vulnerable or non-vulnerable. Same way, every organism, it need not be an infection. In a given condition, a genotype is not diverse enough. It is inviting some trouble for that species survival. Hence, Hydra can follow sexual reproduction also to have variation. But in this fragmentation and budding, that do not happen. In a stable environment, asexual reproduction is effective. When there is no challenge, there is absolutely no challenge, no trouble, everything is in surplus, there is no need of any botheration. So it will do asexual reproduction, and it is effective because the offspring are adapted to that environment. Environment has got a nutrition and it is well adapted. But in an unstable environment, unpredictable environment, what happens? The offsprings, we don't know, you want a diversity. If some good diversity is there, then you will be able to make a living. So you randomly keep looking for a variation. The offsprings may not have the genetic variation to survive. Hence, sexual reproduction is preferred. So, favorable environment, asexual reproduction. Unfavorable environment, sexual reproduction. We will learn more about the Hydra, other species, which will be planaria, in the next class. Thank you.